welcome to Research Bites, your daily dose of science news. A lot of us know someone that's fighting cancer, and most treatments sometimes seem so much worse than the disease. These chemical cocktails of anti-cancer drugs do a real number on the patient's body because they don't just kill off cancerous cells but also damage healthy ones. The side effects are awful for the patient, and their families are often left with a feeling of helplessness. But science has been taking strides towards delivering these nasty but essential cancer drugs straight to the cancerous cell to minimize the damage to the other healthy tissue. In a recent study that was published in the journal ACS Sustainable Chemistry and Engineering, researchers used a protein derived from the serum of cows called bovine serum albumin in a salt solution to make a hydrogel that can act as a drug carrier. So what exactly is a hydrogel? They're jelly-like materials that are mostly liquid but have some solid-like properties. They are formed by a network of synthetic or natural polymers that are dispersed in water. When kept steady, they retain their shape as if they were a solid. Hydrogels that are made with a naturally occurring polymer like a protein are used in medicine because they are not harmful or toxic to living tissue. This means that they are biocompatible. They can also be naturally removed from the body after use. There's a broad range of applications for a hydrogel in drug delivery, tissue engineering, sensing and making artificial organs. The researchers combined bovine serum albumin molecules in saline solution with the help of a cross-linking agent called epichlorohydrin. The crosslinks helped build a gel-like structure that allowed the hydrogel to be highly porous and carry drugs like a capsule. The researchers could also control the size of the pores by controlling the concentration of bovine serum albumin while making the hydrogel. Most gels become liquid when they are subject to pressure or disturbances, like when they are passed through an injection needle or because of the physical stress inside the body. But this hydrogel kept its gel-like structure even after it was passed through an injection needle. And if the hydrogel is to carry drugs, then it needs to retain its shape once it enters the patient's body. And yet again, the gel held its own. The hydrogel could heal itself after mechanical damage and could withstand weights about 300 times its own. The researchers also studied the rate at which the hydrogel released the loaded anti-cancer drugs. They did this by measuring the amount of drug that was released at regular intervals. What they saw was that the drug release continued even after 120 hours. And this ensured the slow and continuous release that was necessary for successful chemotherapy, since burst releases are quite harmful. They found that the drug release is sensitive to acidity, which is actually a boon in disguise, since the pH of cancer cells is also acidic. Besides, the hydrogel is biodegradable, and a digestive enzyme called trypsin that is produced in the small intestines of humans can break it down. This hydrogel is versatile and can carry drugs to treat other conditions besides cancer as well. Other drugs can be loaded on the hydrogel so that it can be used in wound healing and antimicrobial activities. But are these gels ready to be used? Not yet, say the researchers. They have just completed the basic studies exploring the properties of the gel, and now they plan to study it on a living organism. Only when the results of the in vivo studies are positive can they then be used commercially. But since this gel has not been patented, any researcher could use it for further studies. This study brings new hope of cancer treatments that can effectively counter the disease with minimal side effects. Thank you for listening to today's episode. Don't forget to like, share, and follow. This podcast was brought to you by Research Matters, a SciComm initiative by Gubi Labs. For more science-based stories, podcasts, and news, log on to our website www.researchmatters.in.